we heard about bones, but now we are talking about DNA. So, what's your name?
Yesenia.
Hello, Yesenia. Is that spelled correct?
Yes.
Did I say it correctly? So, you are from DNA lab, obviously, and DNA. There are changes on the DNA that you cannot see by sequencing the DNA. So, you're looking at something else.
Yes.
What is it?
So yeah, we're looking at DNA methylation. And,
Can you explain that for kids?
Yes. So sometimes there's these changes in our DNA that can change the expression. So it can turn certain genes on and off. So, but this doesn't change our DNA sequence. We're looking at these small changes to see whether or not we can estimate the age of someone. So
of a living person. So this is not for blood from a dead person or for a pool of blood?
This is blood from a deceased individual.
Ah.
Yes.
How long can you be dead so that your method is still working?
Ooh, good question.
You want to say something?
Yeah, sure. So the samples that we looked at, I think the range of time since death was around 2.5 to 4 years, and so we actually received the samples at a later point too. So
and these were samples on paper, or which types of samples were these? The age samples?
So these were casework samples from the Los Angeles County Department
but
of...
in a tube or on paper?
Oh, these are extracts, DNA extracts. Yes.
Ah, I see.
Yes.
So you received pure DNA, so to speak,
Yes.
swimming in a liquid. Okay, and then that would be the step that you didn't do. Somebody else did that,
Yes.
the DNA extraction.
Exactly.
Then you got the tube, and then what did you do?
So yeah, from here we quantified first to determine how much DNA is there. Then we added chemicals, and that helps convert the bases. So that's part of the methodology. And then after that, we try to determine the amount of converted or non-converted bases in sequences.
By burning the DNA, pyrosequencing.
Right. So that's an interesting name, but it's actually more referring to the amount of light that's produced. And so we measure the amount of light based on the amount of nucleotides added to the sequence of interest.
And could you determine the age, and how close did you get to the actual age of the person, of which you knew the age, right?
Yes.
So you knew the age, so
Yes.
you could test if you were right or wrong.
Yeah, so over here you can see the results. So we had 2 different sets, our training set over here and the test set. And we found that for the training set, the average deviation from the actual age was about 7.6 years. In the test set, it was similar, 7.4 years. And so what we also found is that as the actual chronological age of the donors increased, then the accuracy decreased. So you have the widening and the decrease in accuracy.
Do you think that's due to like bad habits, like no sports, eating fatty food, smoking or something? Or, do you know? Yes, it is.
Yes.
That is so.
Smoking or not maintaining a healthy lifestyle can change your methylation levels.
It can even be changed by stress whilst you're in the womb of your mother, right? So that can also change the methylation pattern. So if you want to be identified correctly, you better eat healthy and you don't smoke. Did you enjoy the work? Was it fun?
Yeah.
How much
definitely.
Like, oh, you're looking. How much fun was it?
It was very fun. It was right before during the pandemic started, 2020, and we were able to push all of this research through.
Oh, you mean you were a lot in the laboratory since you couldn't get out in the streets?
We were locked inside the lab.
Very good. This is the best that can happen to you as a lab worker. So no social contact, just lab work. How long were your hours per day?
Kind of like an 8-hour day, I would say.
Oh, so that was good.
Yeah.
Yours was also an 8-hour day.
Yeah, we were working together.
Yeah.
Okay, so that was okay then.
Yeah.
Very nice results. If you're interested, yeah, just contact the both of you.
Yes.
Thank you very much.
Thank you.
Thank you.